Yes.
Great. Okay, wonderful. All right, thanks again, everyone, for joining today. It is obviously a hard day for some of us in the US, so hopefully I can distract you with some snails today. Okay, sorry, I just want to move the screen stuff out of my way here because I can't see my own. 1 second. There we go. I don't know. Okay. So probably many of you are familiar with this idea that the foundation fauna at deep sea hydrothermal vents host chemosynthetic microbial symbionts. So here's a variety of different invertebrate organisms that we know host very specific microbial symbionts, and that these symbionts are integral to the fitness and survival of these species. So the symbiosis with these microbes in pretty much all of these cases is obligate, and so they absolutely need this microbe for their survival at deep sea vents. And so these animals are hosting bacterial symbionts in or on their tissues that are using the chemical energy that's in the venting fluid. Here you can see a group of animals, snails, clustered around a diffuse flow where there's flow coming out at the top of this video. And this fluid has sulfide, methane, and hydrogen in it that provide energy for the bacteria that are living inside the gill tissue of these snails. And so these symbionts are using the reductants in the venting fluid for energy for chemosynthesis. So they're taking the electron donors that are found in the venting fluid, the hydrogen, the sulfide, and the methane, pairing it with electron acceptors that are found in the surrounding seawater, like oxygen and nitrate, and using that as an energy source to fix inorganic carbon into organic carbon, which is used for their own biosynthesis and growth, but also provides nutrition for the host animal as well. And so you can imagine in these scenarios where this animal has this bacteria that is providing the bulk of its nutrition, it's obligate, that there needs to be some way, some mode of transmitting that bacteria from parent to offspring. If that offspring is going to need the bacteria to survive, then there has to be a way to transmit the symbiont to the offspring. And so one way of doing this is a process called vertical transmission, where the bacterial symbionts are passed to the progeny directly through the germline, actually associated with the germline of the adults, and then grow with the animal as they develop. However, in the vent environment, this is not a very common mode of transmission. There is at least one example, but it's not very common. Instead, what we see more of is this other mode, which we call horizontal or environmental transmission, where the host has a stage where they're aposymbiotic, meaning they don't have their symbiont at some stage. And so the progeny must then acquire the symbiont from the environment, from a pool of free-living symbionts that are either living and persisting in the environment or coming directly from an adult that's around, that's hosting the symbiont. And so this mode appears to be more common in aquatic habitats, including deep sea vents. And this has been reviewed by a nice paper by Shelby Russell in 2019. And so it's thought that one of the reasons for this is that this is a mode that is facilitated by the aqueous medium of water for symbiont transmission, where you have this bacteria or the microbes can persist in the water, in water for this type of transmission process. But this transmission mode has a lot of risks. It has the risk of a progeny not finding their partner at all, and the risk of accidentally being invaded or infected by a cheater or other harmful microbe during the process of taking up a symbiont. And so why would organisms evolve such a risky process of symbiont transmission? And so there's this long-standing paradigm that's meant to explain this. And this is the idea that the risk of horizontal transmission is outweighed by the fitness benefit of associating with a locally adapted habitat-specific bacterial or symbiont strain or species. And so the idea is that you have these progeny that are released into the environment, and when they arrive at a site, they then take up a locally adapted symbiont from that environment. And so this is maybe especially important in symbionts like those at deep sea vents, where the aposymbiotic hosts have to disperse very long distances to new habitats that are variable in their conditions over space and time. And so this is something we see at vents. And so this may explain why this transmission pattern is so prevalent. However, there hasn't been a lot of direct evidence for this idea. And so my lab has been focused on trying to understand these patterns and processes at deep sea hydrothermal vents in the chemosynthetic symbioses that dominate there. And so today I'm going to be talking about some work led by two members of my lab. First, Dr. Corinna Breusing, who's a research associate in my lab, who's focused a lot on the host symbiont population genomics work that I'll present today. And then PhD student Michelle Hauer, who has been focused on understanding the symbionts in their free living stage in the environment. I will also just put in a plug that Michelle has also led an effort to understand how bacteriophage or viruses that infect bacteria might be interacting with symbionts as well, although I won't have a chance to talk about that work today. So we focus in the lab primarily on a group of snails and mussels that dominate communities at Western Pacific hydrothermal vents. And so this is a map of the distribution of the snail and mussel species across the Western Pacific. We have 5 species from the genus Alvinaconcha shown along the bottom there. They're single species from their sister genus Iphomeria nautili, and then a mussel Bathymodial septemdirum. And just to note, all of these species are listed as endangered or vulnerable on the IUCN Red List. And so when we look at these animals living together at deep sea vents in the Western Pacific, we see that in many cases they are found at the same sites, but when we do see them at the same sites, they commonly distribute into these distinct and pretty defined microhabitats, where we have the Alvinaconcha species closest to the vent outflow. At the center of this picture, you can't see it, but there's venting fluid coming out. We then have Iphomeria nautili, the other snail surrounding Alvinaconcha, and then the mussel living at the very edges of these communities. And so you see that there's this gradient in microhabitats where Alvinaconcha is living in environments where there's more chemical reductant available, higher temperatures, whereas the mussels are living in places where sometimes we can't even detect the reductant at all, and they're much lower temperatures. They have these very defined microhabitats. And all of these mollusk species each have their own very specific type of bacteria, either a genus of bacteria or a species of bacteria that are found in their gill tissues. And so this is a phylogenomic tree, sort of modified representative phylogenomic tree of the different bacterial species that are associated with the different animals. And so most of them are from the gamma proteobacteria. One of them is from another group called the Campylobacter rhoda, but each has their own match and pair between the host species and the symbiont, with one exception, which is that there are 2 species of Alvinaconcha that share one of their symbiont types. And so this specificity has been pretty well established by work in my lab, as well as many others for many years on these species. So for example, here, this is a 16S survey, which allows us to understand the microbial populations associated with different animals. And what you can see is that each of these different species, which are shown along the top here, are dominated by 1 or sometimes rarely like 2 types of bacterial species. So that we've shown that this over hundreds of individuals. And so there is very much this strong relationship between species and the specific types of bacteria that they associate with. They really have this sort of well-linked partnership. However, we also know that despite this high specificity and fidelity, within these species, that there is evidence for these being horizontally transmitted, as I was talking about earlier. And so this is, for example, a phylogenomic tree showing the symbiont genomes and the paired host mitochondrial genomes from the species Iphomeria nautili. And since mitochondria are vertically transmitted organelles, if the symbionts were vertically transmitted as well, we would think that those 2 phylogenies would match completely. They would show the same pattern, and they don't. They don't have the same pattern. And this indicates that there's a horizontal transmission mode at work in this species and all the others that I'm discussing today. And another piece of evidence for this is that Michelle Hauer in the lab, in collaboration with Julie Huber's lab, led a study looking at free living symbionts of one of these Alvinaconcha species in the environment. This is a plot showing the sort of composition and structure of the community of symbionts associated with the snails, as well as symbionts in the environment. And what we see is that the symbionts in the environment are very similar to those found in the snails at the same site. And so we're seeing the presence of these bacteria in the environment, and they're very similar to what we find in the snails in that area. So today I'm going to go more deeply into some work that we've been doing, testing the genomic evidence of local adaptation in these bacterial symbionts in one region of the world called the Lau Basin, where we have this series of hydrothermal vents. And so these vents are dominated again by Alvinaconcha, 3 species of Alvinaconcha, Bouchetti, Kojamai, and Strumeri along the bottom there, as well as Iphromyria nautili, another snail, and the mussel Bacomodiolus septemvirum. And so this is a map of just some of the vent fields in the Lau Basin, but those that we are focused on in my lab. And so the great thing about the Lau Basin is it provides this natural laboratory for chemical conditions varying within a pretty small region. And so we find that if you look at the sites along the Eastern Lau Spreading Center, down at the bottom, let's see if I can find my mouse so I can point stuff out. For some reason, it's not letting me see. I don't know what's going on. But along the bottom there, the sort of sites that are clustered together, that's the Eastern Lau Spreading Center, Balufa Ridge vents, and those show a very strong north to south gradient in the concentrations of chemical reductants in the venting fluid, whereas in the north we have higher concentrations of hydrogen and hydrogen sulfide than we do in the southernmost sites. And so this provides a natural laboratory for looking at how symbionts vary and adapt to these different geochemical conditions within one region. And so what we do is that we go to these areas and collect the animals themselves, collect the invertebrates to sample the symbiont containing tissue, which is, in their case, the symbionts are living in their gill tissue. And then we also, in some cases, are able to actually sample the free living microbes in the environment around these animals by filtering the microbes out of the fluid around them to actually see the symbionts while they're out of their hosts as well. And in general, we have this workflow that we use to use population genomics to understand variation in symbionts. And so it's summarized here where we use Illumina metagenomic sequencing of the gill tissue or an environmental sample. We then can use that sequencing data to construct genomes from those metagenomes and create symbiont pangenomes. We also have host transcriptomes as our references. Hopefully soon we'll have some host genomes to work with as well. And so we have these then reference sequences that we can compare all of our reads to. And then we can use those references as a way to assess population level genetic variations according to geography and environment. So we can characterize sequence variation, identify or look for patterns in the presence and absence of genes, or even do tests to try to identify candidate adaptive loci, so genes that are under selection, for example. And so I'll jump right into some results from the host populations in the Lau Basin. And so what you're seeing here is a representation of the genetic diversity and structure of the animals of these different species across the Lau Basin. And so each of these dots is an individual animal, and they're colored by the vent field that they came from. And so what you see is that they're pretty well distributed in these plots, that they're sort of homogeneous. There's no sort of structure by site. So you don't see, for example, all of one site clustering together. And so this shows us that the host animals are not genetically differentiated by the vent field and that they're pretty well mixed. And this ties in nicely to what we know about larval biology, which is not much, although soon will be more through Sean Ariano's work in her lab. But there's a suspicion that these are long dispersing planktotrophic larvae that are able to be in the water column for a long time. And then there are some models that also predict that there are no barriers to dispersal within this basin. And again, so this then fits nicely with this idea that these larvae are dispersing over long distances between these vent fields without barriers. However, when we look at the structure of the bacterial symbionts, according to vent field, we actually see that there are clusters or strains according to each vent field. So again, what you're seeing is individual snails on each of these, or mussels on each of these plots, colored by the vent field that they came from. And what you see is that there's pretty strong partitioning between vent fields. So we see all of the individuals from one vent field clustering together. This varies in the strength of this clustering by host species. So for example, the mussels that you're seeing right in the center of this screen There is some clustering by site, but the variation between the different strains is much lower than what we find in the snails, where the clustering by site is really strong. So this is suggesting, again, that these vent fields each have their own specific strain of symbiont that the animals are associated, that there are these local populations of symbiont that are associating within each animal species. And so we can actually not only see this in terms of sequence variation, which is what I was just showing you, but we can also, again, test to see, are there actually genes that might be showing evidence for selection, that might be showing evidence that there could be adaptation to these local conditions that we see at different vent fields. And so this is some results of a redundancy analysis that can reveal candidate adaptive loci. And what we see is that we find that there are genes under selection that are significantly associated with the fluid chemistry. And those genes, interestingly, are often genes related to interaction with the fluid chemistry, like genes for oxidizing sulfur or for hydrogen. Notably, when we do this same type of test in the muscle symbionts, we again don't see a strong evidence for this local differentiation by site. We see some amount of structuring by site, but we're not finding a lot of evidence that there's these adaptation going on in their populations with the muscle symbionts. And this we think could be related to the fact that the mussels occupy a kind of different habitat than the snails do. The snails are much closer to the venting fluid, and they have this high reductant, high temperature environment. This is likely to be a higher variability environment across different sites. And then also we know that these snails rely less on mixotrophy. They don't really eat other food. Whereas the mussels are sort of in an environment that may be more stable across different vent fields. There's lower variability, and they also do retain some ability to filter feed and to eat other types of food. And so this may mean that the sort of selection pressure for having a locally adapted symbiont is less. So this is our explanation for why we're seeing this pattern in the mussels versus the snails. As I said, one of the other things that we can do with this data is we can actually look to see how genes vary across the different vent fields. So bacteria can actually vary what genes they have quite a bit. And so we actually said, okay, do these different strains actually have different genes? And so, for example, we're looking to see in particular if they have variation in their genes that they need to interact with the fluid chemistry, which seems like an important driver of what we're seeing here. And in fact, yes, we do see variation in their gene content by site. So we see that they either have or are absent with genes that are related, for example, to hydrogen oxidation or sulfide oxidation. We also interestingly see a lot of variation in the genes that they have for interacting with viruses as well. So it seems like there also may be the potential for a local adaptation to be occurring in terms of their interaction with local virus populations too. But I'll go into more detail to explain some of the symbiont gene content and one really interesting and compelling evidence of potentially for local adaptation, and that is the symbionts associated with the snails Alvinaconcha strumeri and Alvinaconcha kojimi, which jointly host one type of symbiont, which we call gamma-1. And so if we look at the populations of this symbiont by site, what we find is that the symbionts associated with the snails in the northern sites, which have higher concentrations of hydrogen, can still have genes for the use of hydrogen as an energy source. So they have the hydrogen oxidation related genes. Whereas if you look at those very same symbiont species in the south, the strains there seem to have lost all of those genes for hydrogen oxidation. And so there seems to be potentially some benefit to having lost those genes if they're not needed anymore. And so we do see, again, this evidence that they're potentially locally adapted to the hydrogen concentrations found at their very specific site. And this, when we discovered this, tied in really nicely to some previous work that we had done actually measuring chemosynthesis associated with hydrogen oxidation in these species. So this was a paper that we had published actually prior to this population genetic study, where we were able to measure how much chemosynthesis was occurring in these species when given hydrogen as their energy source. And we saw a huge amount of variability by site here. So what you're seeing is that here are the rates of chemosynthesis when using hydrogen as the energy source. And we see that each of these colors is a different experiment. And so in the experiment, in some of our experiments, the rates were higher than other experiments. And those experiments varied by the site that we actually took the animals from to conduct the experiment. So when we looked back on this data, we realized that the animals that had the higher rates of autotrophy associated with hydrogen came from the northern sites where the strains should have the genes for hydrogen oxidation, whereas the experiments with the lower rate of chemosynthesis associated with hydrogen oxidation, those came from the southern bend fields where the strains should be missing their hydrogen oxidation genes. So again, we're seeing this, not just the genetic variation, but this is also propagating into their phenotype, meaning like how they actually function. And it does appear that we're seeing differences in their genes that then result in their differences in autotrophy, which is expected, but it's nice to experimentally validate. Okay, so I can summarize so far with what we've seen now is that we are finding and building evidence that these hydrothermal vent mollusks transmit their symbionts horizontally via the environment, and that they associate with geographically specific symbiont strains. And not only are they associating with these geographically specific strains, but that this genetic variation, the strains, the differences that we see in the sites, it can at least partly be explained by the adaptation to local geochemical conditions specific to the site. So this again is sort of reinforcing this paradigm that has existed for many years, that the reason why these organisms are transmitting through the environment, they're risking this, this process by putting their progeny out without their symbiont, that the benefit is that their larvae are arriving at a site and taking up a symbiont that is better adapted to that particular site. And so we are building evidence that this paradigm does appear to be true. Okay, so now I'm going to move into some more recent work that was sort of setting the stage for this particular part of the world and what we have learned there. And now I'm going to go into what will actually end up being kind of a happenstance discovery, but allows us to now look at what happens to local adaptation in the face of local extinction events. When you actually lose a locally adapted population, what happens in that situation? And so you probably are familiar with the record-breaking Hunga volcanic eruption that occurred in January of 2022. This was a stratospheric eruption event, that was the loudest sound, heard in many, many years. It was, sort of globally impacted climate. It's obviously a very important eruption. It also severed communication ties in Tonga, and also, unfortunately, killed a few people during this event as well. And so it was obviously a pretty big event that was fairly known globally. And so these Lau Basin hydrothermal vent sites are fairly close to the Hunga volcano. And so this map here shows in red the Hunga volcano, and then we have some of these Eastern Lao Spreading Center sites, Valufa Ridge sites relative to the volcano, and you can see the plume coverage there. This is the where we actually saw the volcanic plume, and then also some arrows indicating potential submarine flows. So some of the sites that we have been working at were 80 kilometers from the volcano, and others were more like 200 from this volcano, but still obviously fairly close. And so we had been previously scheduled to do a cruise to this part of the world in March and April of 2022, so only a few months after the eruption. And this was a completely by chance event that we were scheduled at this time, but it meant that we were diving on these sites pretty near to this volcano only a few months after this occurrence. And when we arrived at the site, we found a pretty remarkable observation, which is that many of these sites were completely covered in really thick cover of ash. And so here what you're seeing is some ash cover in these environments. You can see there's some white microbial growth that had already started to occur. There's still venting going on at these sites. We see active chimneys, we see diffuse flows. And so you see this microbial growth commonly at these sites. But then you'll see in this video that there were also a lot of empty shells. That's what this, in the sort of forefront there, that there's many of these empty mussel shells that have sort of fallen over there. And so this heavy ash did have some pretty severe consequences for the communities of chemosynthetic animals that live there. And just to give you some perspective on this disturbance event, we were able to compare some photos and videos that were taken on a French cruise in 2019, right before the eruption, or as close to the eruption as anyone else had been there, to our video and footage from our expeditions. You can see some really striking images here. This is the site Tokam, which is very close to the volcano. It's 83 kilometers away, so pretty close. And so what you can see on the left is some devices for collecting larvae that were left out in 2019 for us. And then you can see it tipped over on the right side there in the very same spot. And so there were these sort of vibrant mussel communities around this white PVC collection device that are now no longer there. You just see some ash and some crustaceans. And then another example on the bottom here is just showing some bare basaltic lava pillows. And there's a marker there that had been left on the seafloor that you can see with the yellow arrow. And when we returned to that and found that very same marker, you can see some of the same seafloor features to really match it up. And now we're seeing this incredible cover of ash all over this site. So it was pretty remarkable. Conversely, as we moved towards the south, away from the Hunga volcano, we found that sites away from the volcano had little to no ash on them. And so, for example, this is the site Tui Malila, which is about 200 kilometers from Hunga. And again, we're now matching up to images from the 2019 cruise to a 2022 cruise. And we see that, the markers are shown in orange there that are matching. You can see that the animal populations look fairly similar, and we really could see like no detectable ash at this site. So it seemed like, there were some sites that were unimpacted by the eruption because of being farther away. And so we were actually able to quantify these observations, and this was published this year in Nature Communications, Earth and Environment. If you're interested in seeing the details of this, this is a collaboration with Sean Ariano, as well as Andy Davies from URI and his student Jasper Meager. And so we were actually able to use the footage from 2019 and from 2022 to come up with a semi-quantitative measure of the change in animal abundance before and after the eruption. And what we found is that, so this map you're showing here is one of the vent fields that where we saw the significant change is Tokam. And this was really impacted by ash. And so on the left side, you see all these circles that show sort of the relative abundance of the sort of three main chemosymbiotic mollusks that we're interested in. They're pretty abundant at certain spots, whereas on the right side, you see very few observations of these animals, very, very limited observations of live animals, almost none in some cases. So there's a big change in the chemosymbiotic community due to this eruption. And just to give you an overall summary of what we observed, we saw at the sort of most northern sites closest to the eruption, or I guess I should start out by saying we previously had seen these different mollusk species at these 4 different vent fields in 2019 and before that. But then after the eruption, the most northern site, we saw pretty much a loss completely of species of Alvinaconcha and Ephemeria, the 2 snails, and a very, very small population of the mussels remained. As we moved farther away, going south to the next site, Tahimawana, we saw some small patches of Ephemeria nautili and the mussel. When we went to Abe, which is a little farther south with even less ash, we found that we did see all 3 of these taxa occurring again, although Alvinaconcha was very, very limited in its population size. And then finally, as I showed you at that photo of Tui Malila just now, there was no detectable ash there and pretty much no change in the animal populations there. So we again saw all of these 3 taxa there. So a big change before and after the eruption in the distribution of these animals across these vent fields. And so if you recall, what we found previously is that if you look at the symbions associated with these animals at the different sites, we see that there's these strong structuring or clustering of the symbionts based on their genetic diversity, where we find that each of these sites basically has kind of their own strain of these different bacterial species that the animals associate with. So here I'm showing the Alvinaconca symbionts of the different species of Alvinaconca, and you can see that they're strongly clustered by site. I think we're missing one of the labels here, my apologies, but there should be one that should have been the Tui Melilla, those triangles right there. And so this is a sample set again that we had taken before the eruption of looking at this diversity and seeing this pattern, which we had seen previously. And then we were able to again go back after this eruption and do this all again and collect as many animals as we could from the different sites to look at how sort of population level structure and diversity might have changed in response to the eruption. And sort of unsurprisingly, what we see is that we see the loss basically of these populations from our plots here. So for example, Quillamoana and Tocam, those had their own strains of bacteria, and those strains are no longer around when we look in the animals after the eruption. And that makes sense because their host species were all decimated. So basically what we're seeing is once those host species are gone, then those symbionts are lost from the host associated populations in this whole region. So this is true for Alvinaconca, where we saw a very strong structure of the symbionts across these vent fields, where they each have their own symbiont type. This is also true somewhat for Ephemeria as well, where again, this is what it looked like before the eruption. And then after, we see that loss on the bottom of that, there's that group of Tocam symbionts, that strains of Tocam symbionts are now no longer found in the population. Again, this makes sense because we weren't able to even collect animals from Tocam because the animals had been completely decimated there. And then finally, the mussel, which shows some amount of population structure, but it's sort of a weaker population structure by site. Again, this is what it looked like before the eruption, and then after, fairly similar. So because the mussel was able to survive the eruption better, and then also had overall less differences among sites, we see sort of overall like less of a loss of a particular sort of strain from the overall population in the mussels. We also can use this data to understand the overall genetic diversity of the symbionts and just see how that has changed before and after the eruption. So what you're looking at here are measures of genetic diversity. On the top, the overall genomic diversity, which is nucleotide diversity by site, and then also the genetic diversity as variation between hosts, which is the interhost nucleotide diversity. And so what you can see is that there is a statistically significant decrease in the diversity of the symbionts. This is at the basin scale across all of these sites after the eruption, with the exception of one measure here, which is the mussel symbionts in the sort of overall genomic diversity category. And again, they seem to, because they have a more homogeneous population and they survived the eruption better, we see less of this overall change in their genetic diversity. So this is, again, the symbiont diversity. You may be wondering what happened to the host, and we have all the data for the host as well, but I can at least show you a little bit of it right now, which is here. This is, again, measures of genetic diversity, but now in the host animal, looking at overall genomic diversity as well as heterozygosity, which is another measure of genetic diversity in animals. And so what we're seeing here is that there's no significant decrease in the genetic diversity of the host animals. And again, this makes sense. We knew that their populations were more homogeneous, although significantly affected, but we're not seeing the loss of very specific genotypes through this eruption. And so it does seem like the symbiont diversity has taken a real hit. We're losing some strains from the populations that are sort of no longer found after the eruption, at least associated with the host animals. And so again, just to summarize, what I've just shown you is that these site-specific strains of Alvinaconca and Ephemeria are now no longer present in the regional populations of hosts, resulting in an overall loss of genetic diversity in the symbionts that we don't see the same pattern of in the host animal. So again, we're losing these site-specific strains, at least when we look in the host animals there. But you might be wondering, well, what about the free-living symbiont population? Since these bacteria don't always just live in their host animal, they also live in the environment. So even if we see the loss of these symbionts in the host-associated populations as the animals have died in those regions or in those sites, you might be wondering, well, what about these bacteria as they remain in the environment after the host populations have been decimated? And so we are also working on that as well. And this is sort of newer results led by Michelle Hauer in my lab. And so she led a pretty amazing effort to go out and collect fluid microbial samples across these vent fields using the suspended particle rosette sampler, the super sampler, which allows us to filter approximately 40 liter samples in situ and sort of decide exactly what location we want to be filtering from by positioning a wand to filter water from what we're interested in. And so here's just a collage of different images of us using the super sampler to collect fluid onto filters and collect the microbial populations in those fluids from around the vents. And so you can see that we're, in all these images, we're filtering from around the animal populations or sometimes around the dead animals, where we actually are sticking the intake wand right up against these animals or the dead animals. And then what you're not seeing here is that we also have another set of samples where we're farther away from the vent fields, hundreds or tens of meters from the vents, looking at sort of the background microbial population around these vent fields. And so I'll just quickly summarize what we have seen so far, which is that we can actually still see the symbionts in the environment at these different vent fields. And so we used a method called 16S rRNA gene amplicon sequencing, where we actually include an internal DNA standard that can allow us to back calculate the absolute abundance of the symbionts and their genes in our sample. So we can actually say how many of these genes, and then relate that to potentially numbers of symbionts actually per liter of fluid that we collected. And what you're seeing is for these 3 different symbionts that associate with Alvinaconca, the mussel, as well as the 2 other Alvinaconca species, we see that they are present across all these vent fields that we were able to sample, with a variation between more and less ash. And this is a summarization of the data that includes both the samples we took right near the vents and the animals or the dead animals, as well as away from the vents. And you see that there can be a big variation in the air abundance, but in general, we are finding them, and sometimes even at very high abundances in the vent fields where there's more ash present. And so they do still appear to be in the environment, even though the animals have been, animal populations have been decimated. And so we will be continuing working on this study, trying to better understand the variation within and between vent fields, how this relates to the presence of these animals and the ash, as well as potentially moving more into using also the population genomics to understand how these free-living populations relate to the animal populations at these vent fields as well. So actually looking at, these are definitely the symbionts, but what strains are these symbionts that are present in the environment? Oh, and then the other thing I didn't note, I guess we go back, is you'll see here that I don't have the symbiont of Ephemeria nautili, which is that one snail up here at all. And interestingly, we did not detect the Ephemeria nautili symbiont at all in the environment across many, many samples. And so that's very interesting because it's putatively horizontally transmitted. We expected to find an environmental population of that symbiont, but we didn't find it at all. So that's very intriguing. So just to summarize, the Alvinaconca and the mussel B. septemdirum symbionts are still present in the environment, even after the host populations have been decimated. And so what this means is that the environmental pool of symbionts may still be available to arriving larvae, although we don't know anything about how these symbionts might persist or grow in the environment. So their presence they are present there, and hopefully this means that they might be available to arriving larvae, but we don't know that for sure. So there's obviously a lot here to do in terms of understanding this big disturbance event and what it means for this vent ecosystem now and going forward. And so we do have 2 future cruises that are planned to these sites where we can actually hopefully address some of these questions about recovery and resilience. We're generally interested in recovery in these ecosystems. We will be continuing to monitor the population dynamics of the chemosymbiotic species, as well as other species in these habitats. We're interested in continuing to just survey their genetic diversity and connectivity after this big disturbance, and assess how the symbiont is persisting in the environment, or if it's persisting in the environment, even after this big change in the animal community. And obviously there's many more potential questions that could be addressed here, and I welcome ideas and collaborations from those in the audience. And then finally, I just wanted to note, oops, sorry, there's a weird animation there, that what I showed you today was focused on the Western Pacific hydrothermal systems, and that's where a lot of the work in my lab has been focused for the last few years. But we're really generally interested in trying to understand how these chemosynthetic symbioses vary over space and time, and how they vary across different types of taxa. And so we have some new projects that I want to mention here. We're working on chemosymbiotic from mussels from cold seeps in the Gulf of Mexico and the Atlantic. We're working on the epibionts of the Pompeii worm, as well as some various chemosymbioses from the Galapagos Spreading Center. And we also are interested in welcome new collaborations on your systems of interest. So please reach out if you're interested in collaborating. And so finally, I'll end with thanking my wonderful team of researchers. I mentioned Corinna Broising and Michelle Hauer, who did the bulk of the work that I presented today. I also have some other wonderful PhD students in my lab shown here as well. I always like to thank the Kingdom of Tonga for permission to work in their waters, since everything that you've seen in the Lau Basin is in Tongan waters. Of course, thank you to the vehicles and ship crew that we worked with, and my funding sources. And I will say we also have a completely other side of the lab working on ciliate methanogen symbioses. And so if that's something you're interested in, I am currently looking for a postdoc in that project, and please contact me. So thanks very much for your attention, and I'm happy to take some questions. I'm gonna stop my sharing for now, if that's okay, so I can see people's faces better. Any questions from the audience?
Yes, I have some.
Oh, sure. Hello.
Hello, I'm Pierre Metou from InterRidge.
Yes, nice to meet you.
Yeah, nice to meet you too. Really glad to see your presentation. I'm
Thank
following
you.
the paper from your lab with very much interest each time they go out.
Thank you.
I was wondering about the volcanic eruption and the ash deposition because the Ongatunga volcano is probably like not the only one in the area. And I was wondering, do we have any idea about the frequency of these events historically? Like, is it something relatively common that can happen to this system, or is it something more exceptional?
Yeah, so I mean, we don't know a lot about disturbance in these Western Pacific events broadly. That's a great question about overall volcanic frequency in the region. There are many volcanoes there. Hunga, I think, was a sort of once every 1,000-year type eruption cycle, is what I understand. Yeah, but I will say the limited work that we've done in the Western Pacific, which is primarily driven by Chuck Fisher, looking at sort of how these vents the sort of stability of the vents and the communities over time, which is more on the like 10, 15-year time scale, has shown that they're very stable, at least relative to what we think of when we think of like the Eastern Pacific. But you know, the sort of information that we have is fairly limited. But yeah, that's a good question in terms of their potential for these similar type ash disturbance. I have not heard of anything being documented like this before, either in the Western Pacific or elsewhere for vents. So certainly, at least scientifically, it's not something we've seen much before.
Okay. And I had another question about like the disappearance of some holobiont rather than like the mussel are maintained in all vents, including the one that were the most impacted by ash, contrary to the Biniconcha and Ifremaré is a bit in between. I was wondering, first, like, do you think those are remnant population that survive after the ash, or are they a new colonizer? And then, if they are a new colonizer, is it related to their like differential ability to colonize? Like, Batmolios could be quicker compared to.
Yeah. Yeah. I think they're survivors. So we were there only a few months after, so I think it's unlikely that they, grow at the rate that that would mean that they were normal-sized adults, large adults that we were finding in those impacted sites. So I think it's unlikely that they arrived and grew to that size in the 2-month period. So I think it's more likely that they survived. Sorry, was there a second part of your question?
No, that was
Yeah,
that
yeah,
like are they
yeah.
survivor, and like do you think like some species are more quicker colonizer than other? But I think it would be
Yeah, we do.
interesting to continue with the future cruise.
Abs
On that.
Absolutely. So yes, so I work closely with Sean Ariano from Western Washington University, who's a larval ecologist, and these are some of the questions. She and I worked together on the 2022 expedition, and she has some data about the larvae of these animals, and then also is very interested in their recolonization going forward and the potential for that. Although obviously now these sites are covered in basically sediment, and these animals are not adapted, as far as we know, for settling and developing on sediment. They're usually used to hard substrate. So we'll see what happens going forward, because I think it's obviously a lot of things to learn here.
Okay. And last question, and after I promise I give
Sure.
the audience. Just
Yeah.
out of curiosity, like on the most impacted side, did you see other species than the holobiont that are remaining? Like for instance, like so the non-symbiotic one, like the shrimp, the crab, this kind of thing.
Yes, and so we saw lots of other. So interestingly, it looks like the chemosymbiotic species were the only ones that were definitively impacted by the eruption, which we attribute to their high respiration rate and maybe lack of mobility. We don't really, basically that we think they were covered and then couldn't get out and suffocated, and that chemosymbiotic animals have a very high respiration rate due to their symbionts also needing to use oxygen. So that's what we think. We obviously don't know. But yeah, there were still present populations of crustaceans of many kinds, of other types of organisms, including barnacles or even anemones, and even some like the sort of predatory gastropods, like there's a whelk species there, and we saw that.
Yeah.
And there was no, we saw some variation between the 2019 and 2022 populations, but there was no obvious impact. There was no the way with the chemosymbiotic species, it was very clear that like they were there in abundance and now they're not. The other animals, we just didn't have high enough resolution data to really see any differences, but it was not, so there's no obvious big difference in their populations, even though maybe they were impacted a little bit, but not so significantly that it was that drastic.
Okay. Well, thanks. And yeah, I'm very interested about collaborating in future.
Sure. Yes. Let's definitely talk. Yeah. 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 Hey, Holly, I see your question.
Hi, great talk. So I have a kind of a two-part question. So I'm curious first if do you know if there's coexistence, well, like within a strain for a host? Are there multiple phylotypes sort of coexisting? I don't know if you've tried to use like some of the pangenome tools to look at the phylotypes. And then the second part of the question is whether you have evidence for, you've thought about looking at sort of temporal replacement of symbionts. So if you can kind of have like if these vents are stable over time, whether you can have like replacement of symbionts, either in responses to like environmental conditions or some of the viral dynamics, like if that causes a specific phylotype to go extinct or anything like that.
Yes. Yeah, great question. So yes, we do see that there's a coexisting strains within each animal. So, each animal has it's not a clonal population by any means that they have of that, so within that they usually are dominated by one strain. You can see in our 16S data, which is, low resolution, though, you see like that there's basically one 16S sequence that dominates every individual. Occasionally there's a couple, but it's usually one. When we actually look at the genome, which tells us more about strain level variation, we do see that there are multiple strains there. And so no, your question of replacement in these species, we have no idea if that's something they're possible, whether they are infected early on and then they just sort of grow with their population. Their original infection sort of is what remains throughout their life. We have no idea. It's a great question, but one we don't have good answers to yet.
Great. Yeah, thanks.
Yeah. Yep. Any other questions?
Hello.
Hello.
Hi. Thanks for your presentation. So I'm a seismologist, so I just have a few questions out of my curiosity. So
Sure.
wondering, what are the mechanisms that the ash cause some of the animals go extinct or dead? Like you mentioned, like it might affect the respiration of some of the animals. Are there other mechanisms? And another question is, I'm wondering what the impact of those ash cause the for the hydrothermal system. Are they going to block the system, or yeah?
Yeah, so I'll start with the first one about the animal mechanisms. I mean, I think when animals that are normally, benthic and not covered by ash are suddenly covered by sediment, you see this in like some studies of people who work on dredge, the impacts of dredging, or where you're releasing sediment on animals. They basically can either survive the sort of coverage long enough that somehow it's it's either like basically swept away and they're no longer covered, or they have to be able to get out, they have to escape. And so we think that with these animals, the chemosymbiotic animals, that they couldn't really escape. They just, I guess, physically weren't able to. Even though they are mobile, they can move, but maybe not in the way that would have allowed them to escape sediment. And at the same time, they are breathing, their respiration rates are very high. And so we think that they just suffocated, quickly, and then, or at least more quickly than they were able to escape, if at all. And whereas these other animals, like the crustaceans, are mobile, they may have been able to, they're much more mobile, they maybe were able to sort of escape from the sediment. And then sometimes you would see some of these things like barnacles that were just more on vertical walls, and so therefore weren't necessarily as covered by the ash. And so maybe just their location in the vent field made it that they had less ash cover on them. But it's perplexing because some of these animals that didn't seem to be impacted, like an anemone, you would think would not be happy being covered by ash, but there is definitely something going on there with the differences in their physiology and ecology. The second question about vent fluid flow, we have no idea. I mean, this is where we're really interested in moving forward and understanding how the vent flow could be impacted by the fact that there's now this coverage. We did see there was still fluid flow at the sites, even when there was a lot of ash. There were still active chimneys. There was diffuse vent areas that were covered in ash, but we could either, we actually would stick a probe into the ash and see that it was hotter. And then obviously there was, like in that video I showed, there's some sites where we're covered in microbial mat, indicating that there was still a sulfide source under them, that's venting. So there's still active fluid flow, but now moving forward, I agree that there's potentially, how the fluid is interacting with the ash chemically, the physical potential interaction with the ash coverage. That's all open questions and we have no idea, but we look forward to trying to address in the future. Well, not me, myself, with collaborators.
Thank you.
Yeah.
Okay, well, if there are any last minute questions, now is your chance. Otherwise, I'd just like to thank Roxanne for a wonderful talk. And I'd just like to note this is our last webinar for this calendar year. We'll be back again in January 2025.
Wonderful. Right. Thanks everyone again.
Thank you so much.
Take care, everyone. Bye-bye.
Bye.